Hi, this is Sadashiva from Science Mission. Today we have Dr. Geert Schmid-Schonbein, Distinguished Professor from the Department of Bioengineering at the University of California, San Diego, is with us. Dr. Schmid-Schonbein is a member of National Academy of Engineering and is a fellow of American Heart Association. He is a founding fellow of the American Institute for Medical and Biological Engineering, and winner of the Melville Medal from the American Society for Mechanical Engineering. He won the gold medal by the International Society of Biorheology and Eugene M. Landis Award by the Microcirculatory Society. In addition, he has published several peer-reviewed journal articles, and he has several grants to his credit, and he is in the editorial board of some journals. Welcome, Dr. Schmid-Schonbein.
Welcome. Thank you for having me.
Let's get going now. So can you please talk about yourself, your background, and some of the things you are doing in the lab?
Right. My background is actually, I was a physics student, a physics graduate student, and I studied physics at the time when bioengineering was not academic credit course. But I was very fortunate to be able to get into one of the earliest bioengineering programs in San Diego. Okay. And as a physics major, I became very interested in this topic area on the physics of living cells and that is, in a nutshell, my in life.
Okay. And can you talk about your, how did you progress in your career and how you reached where you are right now?
That's a very interesting question, which in fact in my presentation will become more apparent to you.
Okay.
I basically became interested in the question, is it possible for an engineer or physicist to develop new ideas about the origin of human disease? Not how I started out. I started by studying small blood vessels, blood flow in small blood vessels, cell properties, molecular properties, biological molecular properties. And eventually I found out that these are all interesting basic science problems, but the real important issue is to ask, can you apply them to something that's important for society, and that's human disease? And so today I'm a physicist or a bioengineer who thinks entirely about the origin of human disease.
Okay, that's interesting. And let's talk about your lab people. Can you?
That's the most important part in my life, of course. I had the great fortune of being able to work for many years with a large number of students, undergraduate students, all the way from freshmen to seniors. Many of the students then stayed on and received an advanced degree with me. And not only do we have students from bioengineering, but we also have students from other medical disciplines, which is very important, very inspiring. So I'm going to show you a slide with some of the students that have worked together with me just for the project I'm going to tell you about.
Okay.
There are many more. There are many more. So life in the laboratory basically is a very boring environment, but it becomes very exciting when you have exciting conversations with students, and they are enthusiastic about the problems they are working on.
Okay. So as you brought that up regarding lab is boring, what else other than lab things you and your fellow scientists do?
The most important part is the laboratory.
Okay.
That's difficult to understand for many people, I know. But when I'm not in the laboratory, obviously family is very important. But if you ask me specifically for hobbies, first of all, I like to go hiking. We have wonderful places where you can go hiking here where I live, San Diego. And one of the hobbies I have is to grow things.
Okay.
And at the moment, I'm trying to find out, is it possible in our climate to grow a banana tree?
Okay.
experimenting with that, and I love to experiment.
Okay, that's nice. Nice to know. Coming back to the research, so
Okay.
you have recently have been working on autodigestion and all those things. Can you give us a little bit preview of how come you got interested in that, and then you can go on present your work?
Yes, that I'm delighted to speak about. This is literally my work, my life's work. I'll give you a reason why we're working on this now, but I won't have time to speak about the history of that. And to summarize the history in a nutshell, we made over the years many mistakes, until we finally started to get a focus on human disease that I feel the first time I'm more confident in, as compared to many other discussions in the past. And I hope to tell you why I feel that way. I hope your audience is very skeptical of what I'm going to say.
Mm-hmm.
And I'd love to hear from the audience. Anybody has an objection or thinks this is all not correct, I'd love to hear from them directly and hear skeptical questions. But to answer your question, in my mind, as bioengineer, to work on human disease is one of the most important things we need to do. And here is an opportunity to think about human disease in a fashion that is non-conventional. And most important, of course, is the question, can you open up new opportunities on how to treat disease? And most important, how to prevent it. How to tell your fellow citizen what leads to a particular disease. So this is something that I'm trying to inspire students. And the particular angle that we will take, autodigestion, has come out of many years of, as I said, making mistakes.
Okay. So if you can present your work, that would be great.
And be delighted to do that.
Yeah, I can see that.
Great, wonderful.
Yep.
So you read in this title two things. The top line says digestive enzymes.
Mm-hmm.
So I will be spending a lot of time thinking about speaking about digestive enzymes.
Okay.
And then below is autodigestion. That's a phenomenon, and I will explain what this means. But in today's talk, I will apply it to some diseases, to shock, and to death. These are important problems in medicine. And I need to start out by saying that all this work has been over many years supported by the National Institute of Health, by the American Heart Association, and recently also by a private company. Mm-hmm. And it will become apparent why that is the case. And I have to tell the audience that I have a conflict of interest because I own some stocks in this particular private company, Leading Bioscience.
Makes sense. Okay.
Now, the most important slide I really need to say here already, what we discussed earlier, is what I'm going to tell you is derived from the effort of an entire team of people. On the left side, I have listed students, undergraduate students, graduate students, and postdoctoral fellows who worked with me over the last, especially 20 years on this particular project. Mm-hmm. On the right side, there is a list of collaborators with whom we worked. They are clinicians, they're basic scientists. And so you can think of this idea of autodigestion really being something that has evolved. It wasn't just a single quick idea from a person in the laboratory, but it's something that evolved over many years and many decades for that matter. So a best way to explain why we do and why we focus on the problem that we do is to think about this process that many people in the public know now. It's called inflammation. It's an old word, was introduced by some pathologist about 1822. And it the remarkable thing in the last 20 years, or maybe close to 25 years, is that just about every human disease has now been identified to be an inflammatory disease. We read in the newspapers about that.
Yep.
And I often ask myself, what does this really mean? Those who did studies on tissues, say cultures or animal studies, have known all along that there is inflammation. But it was a remarkable development that clinicians, through clinical trials, were able to show that the patients they were interested and investigating had signs of inflammation. Now, you can roughly divide, this is a little simplified, I recognize that. You can divide the inflammatory diseases into two general classes. One is what's listed here on top, what people call infectious inflammation. These are states of inflammation in patients in which indeed you are able to demonstrate that bacterial, viral, fungi, and other microorganisms may be around. And in fact, there are treatments that medicine has for these particular conditions. There's also another very large class of inflammatory conditions that are, I call them non-infectious. These include, for example, something like acute injuries. This is an injury that happens over a very short period of time. There's not enough time to grow a bacterial culture. Cardiovascular diseases, metabolic syndrome, that is a state of elevated diabetes, arterial hypertension, and so on. Atherosclerosis, it has been repeatedly proposed that atherosclerosis is an infectious disease, but the evidence never became really conclusive, and certainly we don't have any treatment for atherosclerosis today that's derived from an anti-infectious approach. And this list goes on and on. There's cancer, many cancers fall into this, certainly aging. And today I'm going to highlight especially a situation that's called sepsis and physiological shock, and then multi-organ failure. This is sort of a sequence of events that eventually leads to death. And therefore, if you're looking for an important human disease or condition, looking at sepsis is a very good way. And it turns out, as you'll see in a second, this was an important entry point. So when a clinician identifies an inflammatory state, typically in today's diagnostic portfolio, a venous blood sample has been collected, and then inflammatory mediators like C-reactive protein or the interleukin-6 and many others are being measured. And if you have in your bloodstream elevated levels of these particular proteins, then in that particular case, you will be classified as being in an inflamed state. Now, when I look at this inflammatory event, I ask myself, if every human disease has these inflammatory mediators, what is nature telling us? Well, we have to go back to the basic idea of inflammation. It is actually a repair mechanism that nature has introduced, and is unique to living organisms. And this repair mechanism is very nicely studied. For example, if you generate, say, a skin scratch, and you then look at the response of this tissue, that leads into what people call an inflammatory cascade. That's probably a better term. And its whole purpose is to repair. So typical states in there is, right after the injury, an enhanced response in the tissue in terms of elevated permeability. There is ion exchange. There's a variety of other reactions that occur in the tissue that many of these we know in great detail today. These local responses to the injury are followed by a removal of the old tissue. The debris has to be removed. We don't have many capabilities of repairing, as to say, a mechanically injured tissue. It really needs to be completely replaced. And therefore, we have cells like neutrophils and monocytes and lymphocyte infiltration that actually carry out this task. And there's a tremendous amount of work today, aspects of this are understood. That process is then, once the debris has been removed, is replaced by a neogenesis. Both blood vessels as well as tissue is newly generated. And that's, for example, mediated in part by macrophages that start to release growth factors. And these growth factors in turn lead to a growth of blood vessels, connective tissue, and eventually end up with what traditionally has been called the resolution of inflammation. Namely, it is a repair. You may have a scar tissue, but in the end, you can live with it. So this tissue repair is actually absolutely vital in life. We need this not only when we injure ourselves, we need it at childbirth and for many, many other physiological processes. So if you think of inflammation, which is present in all of these patients, you ask yourself the question, if this is a repair mechanism, what has caused the injury in the first place? If this tissue needs to repair, something has injured the tissue. So as an engineer, I ask myself a lot the question, what could possibly injure my tissues? And I wrote here a list of general classes of injury that can occur. Besides the infection, you can, for example, injure your lung by inhaling nanoparticles. You may be subject to some trauma. There are, of course, all kinds of traumas, maybe a rupture of a tissue, puncture, etc. You may expose yourself to elevated temperatures. You may have a chemical exposure. You may be exposed to radiation. So there's a long list of possible mechanisms by which our tissue can be injured. The question is this list complete? And what I will advocate today is no, it's not. And the best way I can explain this to you by illustrating to you a case, a very specific case that for many years played an important role in our work to identify mechanisms for inflammation and for the autodigestion. This is a very simple experiment. All you need to do is to place a catheter into the femoral artery. There's nothing else. Under local anesthesia, you put it in, the animal is anesthetized. And now you reduce the blood pressure. This is shown here as the mean arterial blood pressure from its typical value somewhere between 100 and 110 millimeter mercury. You drop it down by bleeding the animal through that catheter. So now you lower the blood pressure to a value that you wish to have. It's called, let's say, 40 millimeter mercury. You hold this blood pressure for a certain period of time. And at the end, this is in this particular case about 2 hours, you then return also the blood back to the animal. So this is the blood withdrawn at the beginning, and then we return it afterwards. So this is an animal that has been subject to just a very simple process, namely a loss of blood volume for a certain period of time. Injury on the road when somebody's artery is being cut would be the counterpart of what happens in the clinic. And this is called hemorrhagic shock. At the end of this period, you find two types of animal behaviors. One is a group of animals that will just die. Within 2 or 3 hours, they are dead. And then there's another group that will survive. And we were always curious about this, is why would we get two different responses from these animals, even though we applied the same insult in terms of the blood withdrawal. So we spend a lot of time to try to understand what is the difference between these survivors and these non-survivors. And one of the experiments you can do that was instructive is to take a blood sample at the end, just a tiny sample, and you apply it to some naive cells. We happen to select circulating blood cells, leukocytes, that were from a donor. So circulating leukocytes, when you look at them with the scanning electron microscope, are nice and round cells, have these surface folds. Here's another example. And when you take the plasma from the animals that survive, they all look like this. In contrast, if you look at the animals that do not survive and take their plasma, you start to see that a lot of these cells are in an activated state. This is an activation. We understand a lot of details about that, by which the cell starts to project pseudopods. So that's a nice, simple experiment that graduate students and undergraduate students can do. And now ask the important question: what is the difference between the plasma applied to these cells versus the plasma of the non-survivors that generate this type of behavior? So there is something in the plasma of these animals early on, fairly early on, just 1 or 2 hours after the beginning of the underperfusion of the animal, that leads to this particular behavior. And these, by the way, have a lot of consequences for the circulation. They impair the circulation. And there's no surprise that these animals with these type of cells have problems. And this is just one indicator. There are many other cells you could study where you would also find out that the plasma derived from these non-survivors generates all kinds of problems. So I mentioned at the beginning we made lots of mistakes. Well, for many years, we were searching for what could possibly cause this type of response that you see at the bottom. And our thinking was, well, there probably is some chemical in the plasma, some mediator that will stimulate this type of response. And we spent many years, many decades to look for possible candidates. And to make a long story short, it all failed. There were lots of mediators present. For example, a complement we explored for a number of years. Yes, indeed, we find activated complement, but when we did a test, an important test, namely try to remove complement from the plasma of these animals, we did not eliminate this type of behavior. So it was clear that multiple mediators are present, but we were unable to remove these mediators. And by the way, we were not the only ones interested in this question. There were many other groups find all kinds of mediators, but nobody was able to show now that the particular mediator that I had identified, could be lipids, could be proteins, peptides, was actually responsible for this type of stimulation of naive cells. So for many years, I was unhappy because I felt unless we're able to understand how this process comes about, we're not able to make a difference in the survival of these type of animals. And therefore, anybody in shock or sepsis, we would not be able to help. The breakthrough happened when a graduate student, Eric Kistler, one day came to me in the office and said, why don't we look at the different organs of an animal that has been subjected to shock? And I said to Eric, yes, it's an interesting thing, but likely you're just going to find out that every organ could be a potential mediator of these type of inflammatory reactions. And I encouraged him to do that experiment. He took out the different organs that you see listed here, and he incubated them for about 2 hours just on the bench at 37 degrees, just like if it were a reaction in the tissue. Then took the supernatant of these organ reactions and asked the question, which one will activate the cells just like you saw on the previous slide? And out of this experiment came something really surprising. It was predominantly the pancreas. That was a surprise. I said, well, the others have a little bit of that, but the pancreas is really all by itself. I then asked him, why don't you take an important protein that's synthesized in the pancreas, trypsin, I'll explain a little bit more about that, and add this to all the other homogenates to it. And long and behold, he saw that, in fact, trypsin was stimulating all of these cell projections, just like we saw it for the pancreas. In fact, if you add trypsin to the pancreas, you don't change very much. Interesting, in the intestine, if you flush the intestine, which is this blue bar here, then in that particular case, you get very low levels of leukocyte activation. But if you add trypsin back into it, then you get the red bar and you get an elevation that's similar to what you see in other organs. So this experiment, this little in vitro experiment, told us that there is something about digestive enzymes. And therefore, the whole focus of our study suddenly shifted. And I had to think about these organs. So let me take you briefly through it. This is the stomach. Here is the upper part of the small intestine. That's this long organ that's all wound up in our abdomen. And here, right below the stomach, is the pancreas. That pancreas discharges its contents through the duodenum. And therefore, when food comes into the small intestine, right below what's called the pylorus valve, the digestive enzymes and the food that we just consumed will mix and then go on this right through the small intestine. Eventually, it ends up in the large intestine, which is really just one loop. But the digestion of the food will occur in this small intestine, almost exclusively. So what's happening in the pancreas? Well, the pancreas does basically two things in life. One is, and that's a very small part of the intestine, only about 5%, produces the insulin, the islet cells. 95% of the pancreas is a busy organ generating the digestive enzymes. And digestive enzymes, of course, absolutely vital. Otherwise, we could not eat and we could not use the nutrients of what we eat. They consist of 4 classes of enzymes. One are proteolytic enzymes. So they contain enzymes like trypsin, chymotrypsin, elastase, and a few others. It contains lipases. These are lipid-degrading enzymes. Amylase, which can break down sugars. And finally, nucleases, which can break down RNA and DNA. So these enzymes are absolutely vital for life because they are responsible to make sure that when we eat a plate, that within a period of less than 24 hours, this wonderful plate will look like, which I will not describe any further on this public announcement. We're all familiar with this. So we have a machinery in our intestine that we use every day to break down the food that we do, large amounts of biomolecules, as part of our nutrition. This is the digestion part. We take it for granted. We never know that it actually happens. I show you this particular plate because it shows some sausages. And the skin of these sausages is actually made of intestine in most manufacturing of intestines. And you are therefore justified to ask a very important question. It's more than just a dinner talk. What mechanisms do I have to prevent myself digesting my own intestine? What prevents autodigestion? That is suddenly a very important question to ask. Very few people ask that question. Well, what may prevent digestion of my intestine and my intestinal wall is likely a barrier. It's a protection mechanism that's provided by the barrier. And in this electron microscopic section that you see here, this is a cross-section through an intestine of a cat. You see the various layers of the intestine. There is the layer with these villi. This increases the surface area of the intestine. In the lumen itself is the food together with these digestive enzymes I just showed you. This layer is called the mucosal layer, is surrounded by a muscle, 2 muscle layers that provide the peristaltic motion so that a food bolus can be carried along the length of the intestine after a meal. So the question that arises is what protection mechanism is in that wall? Well, you have to magnify it a little bit further. And what I'm showing you here is now one of these villi magnified. And it is layered here in this particular picture, especially clearly visible. Here's the muscle layer that you just showed. This is in a cat. This one is a rat. It looks slightly different. But the basic mechanism by which they produce and absorb and transport food is similar in the two species. These projections are the villi, and they themselves consist of many epithelial cells. So this is a layer of epithelial cells that covers the entire intestine. Now, these epithelial cells have to be protected against your own digestive enzymes. And the way this is achieved is that there is a slimy layer, a mucin layer, that covers these epithelial cells and these villi. And there are two types, actually. There is a mucin 2 isoform, and there's a mucin 13 isoform. The one that 13 actually sits on the epithelial cell, on the what's called the brush border, whereas the mucin 2 is actually fluffed off from a cell called the goblet cell, and it's being carried away in the intestine together with the food. So when you go potty, what you will see is the mucin 2, and the mucin 13 has to stay behind on top of the epithelial cell. Now the question is, okay, if this barrier is responsible for protection against autodigestion, how good is this barrier? We know barriers are not perfect. And when we study that, here's an example of this. We find out that it actually can leak. So I show this to you in these images. They're called zymography. Let me orient you. This is the muscle layer here at the bottom. Up here is the food, and here the villi, barely visible, the villi. What we've done here is we put a little peptide on top of this tissue section, and this peptide has the feature that is essentially non-fluorescent, but when it is being clipped by a digestive enzyme, in this particular case, we targeted trypsin, it becomes fluorescent. So in a sham situation, you see in a normal control, there is a little bit of fluorescence, interesting enough, also on the abdominal side, but it's relatively low. On the other hand, if we introduce what's called SAO, splanchnic artery occlusion, which is nothing else but an underperfusion of the intestine. In that particular case, we start to see this bright blue. If you do it for 30 minutes, in fact, the entire wall of the intestine becomes fluorescent, telling us that now the digestive enzyme, instead of being in the lumen of the intestine, they have actually entered into the wall of the intestine. That is a short period of time, a full entry of digestive enzymes, an escape out of the intestine. Now, the question, is this an important event? Well, this is a bright field image of the same intestine stained with an H&E stain. And you can see this is the normal villi structure. After 15 minutes, the tip of the villi has already been digested away. And after 30 minutes, in fact, the length of the villi is now reduced because the top part has been completely digested. So this is autodigestion. And this is a terrible event. Oh my God. When these digestive enzymes escape, they do all kinds of terrible things. I have nothing else but bad news.
Some problem with the volume. Hello?
Is it going to anything? Is this better?
Yeah.
Can you hear me?
Yeah, now I can hear. Go ahead.
Yeah. It's usually very low. The black bars is the protease I showed earlier. So
just breaking again.
Is this on my part?
Hello?
Yeah?
Yeah, go ahead. Now, let's see.
Okay, I hear you very clearly. Can you hear me?
Yeah. In between, it was kind of like blurry. Yeah, again it is blurring. Hello?
Hello?
Yeah. Is the microphone near your mouth or so?
Everything is just normal, like anything.
Okay. Go ahead.
So she finds plasma proteins in these different plasma proteins. Also finds gravity.
No, it is breaking up. Lots of it.
Should we reestablish a line? Because I don't know what to touch here on my computer.
Okay, no, it comes out now, okay, but when you start going, it is kind of like breaking up really.
Is it better now?
Yeah, it is better now.
First, you have to tell me where you...
Is it better now?
Yeah, go ahead.
So we find that
no, now again it is breaking up.
Okay.
Hello? I can't hear you properly.
Are you back? Would that be okay?
Just mute and unmute your this thing once, just to make sure that it works.
And again. More time to do this?
No, it is just kind of
reception.
Okay, I don't hear you. Just talk something like so that I can make sure that you can hear me, right?
1, 2, 3, 1, 2, 3.
No, yours is totally breaking up.
Should I use it?
Okay.
Should I connect Google Hangout?
No, if you quit, what happens is it will go away. Everything will go away. Because it's recording. So let me see.
1, 2, 3. 1, 2, 3.
Okay, it is fine now.
1, 2, 3. Okay.
Yeah, I can hear now.
Is it better?
Yeah.
Yeah. We had some rain here in San Diego, so but it just stopped.
Okay, now I can hear you. It is fine now.
Okay. So can you edit this then later on?
No, I can't do anything. I have no option. Which is okay. Which is okay. It's about 20-30 seconds. That's okay.
Okay. So both the plasma and the peritoneal cavity are filling with digestive enzymes after hemorrhagic shock. Okay. And these enzymes are actually active. They are not just present as a protein, but these are active proteases. There is a second effect that takes place. The digestive enzymes will activate other pro-MMPs and other pro-enzymes. And this is an example for MMP9. Both the molecule itself is not present before shock, but it appears after shock. And this is shown here by a zymogram and by a Western blot. And these are the measurements associated with that. We also find that digestive enzymes, besides being in the plasma, are also present in the lung, in the liver, and in the heart, and even in the brain. This all within a period of about 2 hours after hemorrhagic shock. So this evidence tells us that digestive enzymes are escaping from the lumen of the intestine in hemorrhagic shock, and they escape into just about every organ in the body. Now, the question arises, yeah, maybe they're not doing very much. Well, I can give you one example that illustrates in a very dramatic way that this entry of digestive enzymes is a highly destructive process. And it's illustrated by this diagram. That's a mass spectral scan, somewhere between about 5,000 kilodalton and about 15,000 kilodalton. And the peaks you see here are typical biomolecules that are, many of them are well understood to be present in the plasma of a rat. Humans look similar. These molecules are vital signs used for all kinds of vital processes. And this particular profile, I could show you many others, they look very similar, is before the animal is in shock. So this is the profile that you would see in a healthy animal. If you now go from the same animal 2 hours later after shock, look at the same spectrum, it looks like the one here at the bottom. And the best I can describe this is there is a wholesale destruction of biomolecules in the plasma of these animals, and therefore no surprise that these animals cannot survive. We carried out a more detailed study where we looked at the peptides present. These are peptides between about 5 amino acids and 10 kilodalton. And whereas a control animal has about 126 such peptides, after hemorrhagic shock, this goes up almost by a factor 2 to 3. So these animals are destroying their own biomolecules due to the presence of these digestive enzymes. These biomolecules come from many different sources, and we have been able to identify several of them. In fact, they are published. One important question that came up early in this work was, okay, this shows a schematic stomach, intestine, and colon. If this process occurs in the small intestine, would there ever be a possibility that we can interfere with those digestive enzymes? And the approach we developed early on, this was in 2000, is to say, why don't we make local injections into the small intestine? We introduce an inhibitor for the digestive enzymes. We used a drug that was previously developed to inhibit digestive enzymes in the case of pancreatitis. And we were able to show when you do that, you block indeed digestive enzymes. You block it, go back one more time, in the small intestine only. We didn't touch the large intestine, neither did we touch the stomach, just over this length. We found out that if you do that and you expose the animal to hemorrhagic shock, and you then wait for an extended period of time whether the animal will survive or not survive. This is a group of animals, 12 animals. 3 only survived because they were not treated. In the animals that were treated with this drug I mentioned, ANGD, and it was given enterally, that means just in the lumen of the intestine, the treatment group had 10 out of 12. We then said, well, maybe this is some artifact about this particular protease inhibitor, which is a synthetic molecule developed by a Japanese company. Why don't we use some other molecules? We used another protease inhibitor called tranexamic acid, and an older one that's been in the literature for a while called aprotinin. They have very different chemical structure. And we repeated these experiments by administering them into the lumen of the intestine enterally. And we're able to show that you get a significant increase in survival in all of them. As a matter of fact, we were able to show that in other forms of shock, other than hemorrhagic shock, you can get a similar improvement of survival. What does this look like in real inside an animal? Well, this is a normal intestine of a rat. This is the right heart, the atrium, and here's the lung. This is how it looks like in a control animal. This is how it looks like in an animal that has been subject to this hemorrhagic shock. The dark red coloration you see is due to the fact that red blood cells were escaping across mostly venules in the microcirculation, and they leak directly into the tissue. So you can't wash them out. And this leakage or lesion sites develop in the right heart, in the lung. Every one of the animals looks slightly different, but this is a consistent finding. When we block the digestive enzymes in the lumen of the intestine, so we don't give any drug IV, we just block them right in here. And this is after 1 hour. So in other words, we were trying to simulate a human situation where it takes about an hour for the patient to be delivered into the intensive care unit. Then we start to see that there is a significant improvement of these lesion formations, both in the heart as well as in the lung. So this told us if we block these digestive enzymes at a site where they're very concentrated, we can potentially interfere with this, what's called multi-organ failure, that is the consequence, or the manifestation of which we see in these particular images. In fact, over the years, we have done lots of studies with this idea of blocking digestive enzymes directly in the lumen of intestine. Indeed, the animals cannot digest during this period of time. And here I want to show one other example of how this works. The animals develop what's called an acute insulin resistance. Let me explain. So if you give an animal a bolus of glucose, you start to see that it takes, in these cases, about 2 hours for this glucose in the blood to return back to control value. You can also do the experiment in an inverted fashion. You give insulin, so you see a drop in glucose levels, and gradually it restores again. This is how a control, healthy rat would respond. If you now take an animal that has been subject to hemorrhagic shock, these are the red points, you start to see that, first of all, there's already an elevation of the glucose levels, even in the early stage. And then, in fact, the animal really never fully recovers its blood glucose levels. If you give insulin, you get still a response and a reduction of blood glucose levels, but it is a sluggish response and much reduced. If you treat the animal, these are the blue points, with an inhibition of digestive enzymes, just like I showed you previously, you see that this hypoglycemic or insulin resistant state is in fact normalized back to a control. This experiment taught us that the digestive enzyme do damage to the insulin system. And the way we examine this is by taking an antibody against the extracellular domain of the insulin receptor. So here you see some blood vessels in the microcirculation, arterioles, venules, and capillaries that have been labeled with an antibody against the extracellular domain of the insulin receptor. And this is how it looks like on the endothelial cell. If you do the same labeling, identical optical conditions, of an animal that's in shock, hemorrhagic shock, you see still some labeling. But if you compare this image with that image, I hope you can see that on your screen, you see a dramatic reduction of the insulin receptor density. If you pre-treat the animal with tranexamic acid, in this particular case, this loss of the insulin receptor labeling on the extracellular domain is not present anymore. That's supported by direct fluorescent measurements. These are digital fluorescent measurements, both in the arterioles, capillaries, and venules. By the time we treat, we attenuate this loss of the insulin receptor. So the concept that became apparent as far as the receptors are concerned is that a normal insulin receptor, it's a dimer, very well understood today. When insulin comes around and binds to the extracellular binding domain and then signals internally towards what is eventually a glucose transport through some glucose transporters into the cell interior, That process in shock is interrupted, and it's interrupted at the very beginning, namely proteases that are coming from the extracellular compartment, digestive enzymes included. They will clip the extracellular domain of the insulin receptor. So the signaling mechanism is now interrupted, and as a consequence, all signaling that is downstream of the insulin receptor transport chain is now interrupted, and you have an insulin resistant state. We're able to show this phenomenon in a variety of different receptors over the years, and therefore it is this loss of cell function is an early stage of autodigestion by the digestive enzymes. Now, a very important question came up early is, okay, if inhibition of digestive enzymes in the intestine would help, could it ever make a difference in a human being? And it turned out that I was witnessing a case, a special case, which I want to show you briefly. Now, how do we introduce into humans digestive enzymes? Well, the only way we can do that is we can't do it like we did it in the rat, where we had to open up the abdomen. We have to do it with a catheter. And what we do is we place a catheter directly into the most distal part of the stomach. It's difficult to go any further. And then we drip in a protease inhibitor. And that protease inhibitor will slowly make its way through the rest of the intestine. So this process took place in a patient in a heart transplant center that had a heart transplant, and afterwards, a couple of years later, had a motorcycle accident. And this individual, by the time we came in contact with, was in a classical state of shock and sepsis. The intestine, when looked at with an X-ray, was totally distended. And when you looked at the biochemical parameters and the hemodynamic parameters of this patient, it was so that the patient would not have survived even a few more days. So the clinicians at the time, based on consent by asking the loved ones, decided to place a catheter just like I showed you. They treated this patient. This was the first one, to our knowledge, in the medical history, in the medical literature, and they treated this, and this is the same individual intestine 16 days later. This was at the time he was transferred out of the ICU into the hospital ward, and this individual about 3 or 4 months later, in fact, went back home and is still alive today.
Wow.
What is unusual about this case is because this person had a heart transplant, he was not only not quite so healthy as a normal individual, but on top of that, he was immune suppressed, and he responded to this. So this idea is today in clinical trial. We are in the process of enrolling a group of patients that have such complications. And we are hoping that by the end of 2019, we will have the first results, whether this type of treatment could potentially help. And by the way, this is, in order to do these type of studies, they're very expensive. You need to have a company that does that. And that's what this company I mentioned at the beginning, Leading Bioscience, is actually doing at this point. Now, another independent piece of evidence became available to us just a few months ago. In a consortium study with several European trauma centers, we were able to get plasma from septic patients. These are very sick patients in that. And we were able to get it over 2 days, on the time of the day they are delivered and the day later. And it's a small group, only about 23 patients. But what we did with this plasma, we reasoned in the following way. We said, well, if digestive enzymes are present, just like we saw it in the rat, is there a possibility that the patient's own proteins are being broken down into individual peptides? So we did mass spectrometry. We counted peptides in a certain molecular weight range. And you see here a measure of peptide abundance for the case of septic patients. These numbers are from individuals that would all survive. If you look at individuals, there's always a small proportion. In our case was 6 patients that would not survive. They already had, on time of delivery, elevated levels of these peptides. But on the next day, and this is in many of these patients just sometimes hours or just a few days away from death, we saw this dramatic increase in peptide abundance, telling us that these patients, just like in our rats, were destroying their own digestive enzymes. We could get some bioinformatics information about what proteases may be involved in that, but we don't know. We don't have independent measurements of the actual proteases. We suspect that chymotrypsin is a major player. And these values are dramatically higher compared to individuals like controls or healthy individuals. So this is, to us, an independent signal that when digestive enzymes get into the bloodstream, they will actually destroy not only receptors, as I showed you, but they also destroy plasma proteins. So in summary, what I've told you up to this point is that this escape of digestive enzymes activates secondary enzymes. I showed you an example for the MMPs. It leads to an actual destruction of the tissue. We just call it autodigestion. There's receptor cleavage. Many different receptors are involved. And eventually, if the case is severe enough, it leads to multi-organ failure and death. And if you inhibit the digestive enzymes through this enteral administration, you can actually interfere with all of these processes. You don't interfere with the escape of the digestive enzyme because that's a breakdown of the barrier, but you can interfere with all of the secondary reactions that take place. And so today, the big question is, could that be used to save the life of patients in the intensive care unit, patients with sepsis and so on? The good news is the clinical trials are underway, and we have to wait for the outcome of that. So what I told you so far is this digestive enzymes can kill you. If they are allowed to escape, you don't need to enrich them. You don't need to have a higher expression of their values. The ones you have in your intestine is enough. So if the barrier breaks down, it can kill you. Given this fact, I was curious for many years, well, okay, in shock, there is this dramatic breakdown of the barrier I showed you. But what about more milder circumstances? And I had the great fortune to being able, with a group of graduate students and postdocs, to study an animal which is known as the spontaneously hypertensive rat. I'm going to use the acronym SHR. This animal is celebrated today, was generated by Professor Okamoto at Kyoto University for the fact that its mean arterial blood pressure of its lifetime, this is a 12-month record that you see here, is significantly elevated compared to an animal that is called the Wistar Kyoto rat. That's a low pressure control animal compared to the SHR. So this animal has, over its entire lifetime, an elevated blood pressure. The NIH spent many, many millions of dollars to understand why this animal has this spontaneous hypertension. I've had the fortune, as I mentioned, to study this with our students in the laboratory. And what became apparent over time is that we were looking at the blood pressure as a symptom. In reality, this animal was developing a lot of other complications. It has an unusual endothelial apoptosis. It has insulin resistance, just like I showed you before. It has a defective mechanotransduction mechanism. You can study various cells, endothelial cells, white cells, they all have a mechanotransduction mechanism. But in these cells, it's defective. It's not gone, but it's defective. You have an immune suppression in these. The animals can't sleep very well. They have insomnia. And in fact, if you study various cells, you find out that just about every cell you put your hands on has a defect in this particular animal. So it became clear to me that this animal is a representative, is one example, it's a genetic model of what we call the metabolic syndrome. In other words, there is more than just an elevated blood glucose level or an elevated blood pressure that is present. And there's an underlying mechanism that could lead to a complication that extends not just one particular symptom, but many others. And with that, we went out with this idea that there may be uncontrolled proteases and started to ask the question, can we find proteases in the bloodstream? And the short answer to that, yes, we found it. When we just take plasma and we measured with a very broad-acting casein substrate, the activity, we can see that the SHR has elevated levels compared to this normotensive control. And by the way, both of these strains of animals have an elevated level compared to the wild type from which they were originally derived. We spent a good deal of time asking the question, which proteases are activated? So we found out that MMP2, MMP9, MMP7 were activated, others were not. And it was always sort of a mild elevation. It wasn't by 3 orders of magnitude or so. It was a 20–30% elevation. But it was active. And that raised then the question, is there a possibility that there is a chronic receptor cleavage going on? So we decided, let's look at an important receptor called the beta-2 adrenergic receptor. It sits on the arteries, smooth muscle, and it this particular guy is responsible for a vasoconstriction vasodilation when you introduce a neurotransmitter like epinephrine. So if that comes in the bloodstream and hits this adrenergic receptor, it's a very well studied receptor by the pharmacologist, you can see that in fact the blood vessels should dilate. In the SHR, it doesn't do so. And when we, by the way, this is a crystallography of the receptor. When we purchased and found an antibody against the extracellular domain, the so-called N-terminus, and we labeled, this happens to be the aorta, the outside, the label gives you the dark brown color that you see here. So the dark brown color indicates to us that on the Wista, on the WikiRat, there is some labeling present. When we use the identical method, this is all based on a very careful standardization of procedures, and we labeled an SHR endothelial cell. This is the labeling we found. We measured this out with fluorescent measurement techniques, telling us that this particular receptor is no longer present in its intact form to the same degree as it is in animals with controlled blood pressure. When we used an antibody against the C-terminus of the beta-2 adrenergic receptor, we don't see this phenomenon. The inside of the SHR is labeled as well as the outside. And that tells us that, in fact, something on the outside here where our antibody binds is destroyed in the SHR. To make a long story short, we became interested to look at other receptors in the same animal. This is a labeling of the insulin receptor. So here on this endothelial cell, there's a magnified view of an arteriole there. You see this little dark line, this very thin line. It is absent in the SHR to a large degree. It's a little bit present, but much less than in the wiki. When we treated these animals with a MMP inhibitor, I remember I showed you this elevation of the MMP inhibitor, and we used over the years actually multiple MMP inhibitors. This is a treatment that you need to administer over a couple of weeks. And then you start to see that, in fact, this insulin receptor labeling, I show you right here, it comes back. And the animal restores that very significantly. Question is, does this make a difference for clinical parameters in these animals? So here's the mean arterial blood pressure. This is elevated in the SHR. It's about 174 millimeter mercury. When we treat this, this is one of the treatments group. We used a chemical called doxycycline that's at low concentration, an MP inhibitor, at high concentrations, an antibiotic. We find out that its blood pressure comes pretty much down to the value of the control animal. But even in the low pressure control, we find a reduction of the blood pressure on that. For the same treatment, we now look for the blood glucose level. I mentioned to you this animal has insulin resistance. So it has a slightly, it's not a dramatic elevation, but a slight elevation of the blood glucose level. This is glycated hemoglobin value of 5.7%. When we treat the animal, same treatment as up here, we bring this elevated glucose level down to controls. So this was the first direct evidence that, in fact, a single treatment that gets at the root cause of the problem in these animals, namely the cleavage of receptors, is addressing both aspects. Well, once this idea was born, we immediately became interested in other types of receptors. So let me show you one or two other examples. The SHR is very well known for a phenomenon called capillary rarefaction. This was originally discovered by some clinicians, and they pointed to the fact that in various places where you can see human capillaries, for example, in the eye or in on the skin, the patients with elevated blood pressure had a very poor capillary network. It's rarefied. And our SHR has the same situation. This is a magnified view. It's just a flimsy, small capillary network, and compared to the wiki. When we treat this animal, this is the same treatment I just showed you with doxycycline for a couple of weeks. Then in that particular case, that capillary network starts to regrow again. By the way, it doesn't quite regrow to the same degree as in the wiki. So there are still some differences, but nevertheless, it's a dramatic growth compared to the untreated animals. So this capillary rarefaction is due to the cleavage of the vascular endothelial growth factor receptor. If you treat an animal against the VEGFR2, it's called, you can achieve, in fact, the same reduction of capillaries in the tumor field. This is used as a treatment to alleviate. So our concept that I showed you earlier for the insulin receptor, if you look for the beta-adrenergic receptor, which is responsible for arterial vasodilation and reduction of blood pressure, or you look at the vascular endothelial growth factor 2, needs to be applied now for all of three of them. And the key culprit here for a dysfunction of these receptors is that you have this extracellular community of proteases that are clipping these receptors, destroying them. And as a result, their normal function, like vasodilation, normal glucose transport, or endothelial survival, is interrupted, and you get these clinical signs. So this evidence became and strengthened our argument that, in fact, the escape of digestive enzymes and the generation of secondary proteases can lead to widespread organ dysfunctions. And we have studied this in the meantime for many other receptors, and we find the same circumstance. They are being clipped by digestive enzymes or secondary enzymes, and they are responsible, therefore, for a lot of the cell dysfunctions. If you treat these animals with one single protease inhibition, you can actually interfere with all of these what's called comorbidities. One of the questions that arises is, would this ever apply to humans? Well, just very recently, and this is still in press, should be coming out very shortly, we asked the question, is there ever a possibility that digestive enzymes can appear in the bloodstream of humans? So we decided, together with some graduate students, to use a newly developed technique by Professor Mike Heller that allows us to measure MMP2 activity, elastase activity, trypsin activity in addition to blood glucose level, in an individual that just had a meal. So these volunteers ate a meal, sometimes in the morning, and it was a meal that contained, besides a hamburger, hot chocolate, orange juice, and some donut. So it's not a particularly healthy meal that they ate, but we were curious if we were to follow them over a period of time, we decided up to 5 hours, what would their blood glucose, their protease activity looks like. And this was done with a new technique that measured the blood glucose activity, the protease activity in much greater, with much greater accuracy than traditional methods. And the method is described and in the literature, and we have applied it to a variety of different proteases at this stage. What we see there is really interesting. This is the light blue curve is when the volunteer didn't eat anything. This dark blue curve shows you almost a 50-60% increase in protease activity just right after the meal. The meal takes about 10-12 minutes to finish, and within the next half hour do we see this elevated protease activity. So just eating a normal high-fat diet meal is enough, according to these data, to increase your protease activity. It doesn't make a difference which one you choose. It then quickly returns down again, fluctuates. We don't understand why it fluctuates, but it does. It also goes hand in hand with an elevated blood glucose level. When you do the same study with volunteers who are pre-diabetic or fully diabetic, you see that a similar phenomenon, but all the curves are much more elevated, and it takes longer periods of time for them to come back down again. So this is an indication that there is, and this is really surprising, as we go through life. By the way, these are young college students here. Even in those individuals, it is possible that we find digestive enzymes in their bloodstream during a normal day. This process, you may argue, well, you discover that because you used a substrate to see so. And we quickly said, well, other than cleavage of a substrate, why don't we look at something much more interesting? So Augusta Modestino, who did these studies, decided, I'm going to take some blood samples, blood cells of myself. So we're looking at Augusta's white blood cells over the course of this experiment. And what she did is took the plasma from the volunteers or the plasma from the patients and applied it to her own cells, and then labeled her cells. She applied it typically for about 2 hours. And then she labeled, just like we have showed you previously, she showed the insulin receptor labeling. So it took an antibody against the extracellular domain of the insulin receptor, and then used immunohistochemistry. So a normal insulin receptor is what you see in this particular cell here. If you do this to a cell after such a meal, after about 45 minutes, do we see that almost no labeling exists anymore. She did it on many hundreds of cells, and these are some measurements. So she's able to show, while the blood glucose level is just elevation, it's starting to progress to fall down, she sees that there is a large cleavage of digestive enzymes, by digestive enzymes of the insulin receptor. So what we saw in animals, suddenly we see also in humans. She did this, this is a control experiment, so these individuals did not eat anything. We don't see the cleavage of the receptors. The label density is pretty much the same throughout the course of this experiment. When we do this with pre-diabetic or diabetic, we see a dramatic cleavage of the insulin receptor. These are her cells, but exposed to the plasma of these patients. And there is a dramatic drop-off. So this is the first indication that, in fact, this phenomenon of receptor cleavage also occurs in humans. in fact, in young college college students. And this is a process that needs much more investigation and is potentially an explanation for the insulin resistance that can develop under these circumstances. So what I told you today is in a long-winded fashion, that besides injury mechanism that can lead to inflammation, like infection, trauma, and so on, there is an additional mechanism that's present, and that's autodigestion. And it's merely an escape of digestive enzymes out of the intestine. The normal enzymes that are there to digest our food will actually digest our own tissue. So I'm at this particular point at the end of this story. There are many ramifications of that. I thank you all, and I will always end up to wishing my audience a good life and something that they probably never thought of, namely, I wish them a very slow autodigestion. So we will have to see where this story goes. But that's the end of mine for today.
Thank you so much, Dr. Schmid-Schonbein. So if you can...
Should close off this window?
No. You need to just do the... Yeah, you need to just unshare it. Yeah, here you go. So it was very interesting. So I never heard of this, but it really like asked so many questions. Like I'm thinking about so many questions right now, basically, in terms of various diseases and all those things. We can discuss eventually. Maybe just a couple of questions, because we really went through in detail of all the things. So like you had used MMP for some of the diseases there, like to stop the digestive enzyme cleavage. So is it a universal, or like it may be different for different diseases, or like you have some ways to figure out, hey, what is good for this, what is good for that?
Right, that's a wonderful question. We will, the short answer is we don't know. I think we have techniques today by which for different diseases we can find out what are the proteases actually that are active, that are present, that are active, and what are they doing. I think that will open up a very direct line of research, for example, in the diabetic patients, that tells us, okay, they have certain trypsin, chymotrypsin, whatever the case may be, that are under outside of control, and we can find out and find the most optimal way to inhibit this. This is at the heart of this story. In my mind, it simplifies the thinking about many diseases a great deal. You notice that I never spoke about gene expression, because all of these events are happening and are initiated by extracellular proteases. They are the culprit. And my feeling is, rather than thinking about what genes are expressed or how can I correct a signaling pathway, my thinking as an engineer is very simple. If there are extracellular proteases that cause this type of cellular dysfunction, the first thing I want to do when I think about intervention for patients, how do I stop these proteases? So we think a lot in the laboratory about these questions, and we're developing techniques that allows us in specific disease situations to go directly at these. Sometimes you're lucky that you're able to detect these in plasma, and sometimes it's so that the digestive enzymes, I showed you examples of that, are actually sitting in the tissue. So venous blood samples may not tell you the full story.
Okay, and this is kind of getting into really personalized medicine kind of thing. Like, okay, you take everybody's like patients, like for particular person, it may be a little different than the other proteins, which are getting digested more depending on their situation. Right. So you can basically give particular dose of more of this inhibitor than the other. So like, since you have this company, so I was just wondering, are you planning on doing any diagnostic kits, kind of like tools to do this?
Right. Mike Heller, Augusta Modestino, who have worked extensively on this particular one, are the ones who actually would like to start a company. Yes.
Okay.
And Mike's idea is that he would like to have a manifold, so that with a single finger prick blood, we will be able to determine in a particular individual at any instant of time which proteases are actually present. And then we develop inhibitors that this particular individual can take to reduce this activity.
That would be really, really interesting. So yeah, it brings in together diagnostic plus like the other company where you are trying to cure the diseases. So it's a really nice work. Really, I appreciate your contribution to this field for last few decades, basically, and reaching to the point where really you can look into the humans right now to solve some of the problems. And your example was really fitting to the real what is happening these days, like where you don't have any solution to, like, for example, the sepsis and all. So this tool, this way of thinking might be helping them to the doctors to have something to treat the patients. Thank you very much. I will be sharing this with everybody on my LinkedIn, Facebook, and you'll be able to get some response from the people. I'll give you, if it is okay, I can give you your email address, right?
Yeah, please do so. Thank you
Yeah,
for the opportunity, and I look forward to the discussion.
sure, certainly. Thank you so much. We'll
So
get
long.
off from this right now.